Welcome to Computational Biology. Today, we are diving into one of the most fundamental and powerful tools in the entire field, sequence alignment. Imagine trying to find the differences between two massive editions of the same book. You would line them up word by word. In biology, we do exactly this with DNA, RNA, and proteins to uncover evolutionary secrets, discover new drugs, and understand genetic diseases. Let us begin our journey into how computers read the language of life. Now that we know what our goal is, let us look at what sequence alignment actually means. At its core, it is the computational process of arranging two or more biological sequences to identify regions of similarity. Why does this matter? Because similarities often indicate functional or evolutionary relationships. When sequences do not match perfectly, algorithms insert gaps, represented by dashes, to align characters conserved over time. The mathematical goal is simple: maximize the matching characters while minimizing penalties for mismatches and gaps. If scientists find a new human disease gene, they align it with animal databases. Finding a match in mice, for example, allows them to test treatments. This is the engine driving modern genetics. Building on the idea of lining up sequences, let us explore the simplest way to visualize this: the dot matrix method. Think of it like playing a game of Battleship. We write one sequence across the top and another down the side, creating a two-dimensional grid. We then scan the grid and place a dot wherever the row and column characters perfectly match. When two sequences share a long region of similarity, these individual dots connect to form continuous diagonal lines. This method is incredibly useful because it gives us a quick bird's-eye view of the relationship between two sequences. It easily reveals large-scale structural changes, like insertions or inverted repeats, making it a perfect preliminary step before heavy computation. To understand how a computer generates this grid, we need to define it mathematically. The formula is a simple conditional statement. The term d of I, j represents the numerical value at a specific row and column in our grid. S1 of I is the character in the first sequence, and S2 of j is the character in the second sequence. If these two characters are identical, the function outputs a 1, instructing the computer to draw a visible dot. If they are different, it outputs a 0, leaving the cell blank. As the sequences get longer, the grid expands, but this simple binary logic remains exactly the same. Let us see this formula in action with a concrete example. We want to align the word hello with the word jello. We place hello across the top and jello down the side. Starting with the first row, the letter J, we compare it against every letter in hello. There are no matches, so the first row remains completely blank. Moving to the second row, the letter E matches the E in hello, so we place a dot at coordinate 2, 2. We continue this exact process for the remaining letters. The final result is a continuous diagonal line starting from the second row and column. This visually proves that the ending sequence, ELLO, is perfectly conserved. Here is exactly what that completed grid looks like. Notice the letters H, E, L, L, O across the top axis and J, E, L, L, O down the vertical axis. Look at the first row and the first column. They're completely empty, which immediately tells us there is a mismatch between the starting letters H and J. But look at the rest of the grid. You can clearly see the solid dots forming a perfect diagonal line from the top left towards the bottom right. This diagonal line is the visual signature of a conserved sequence. The human eye is incredibly good at picking out these patterns, which translates directly to finding conserved genes in biology. While the dot matrix is great for visualization, we need rigorous math for deep analysis. This brings us to dynamic programming, specifically the Needleman-Wunsch algorithm for global alignment. Imagine trying to find the best path through a maze. Instead of guessing the whole path, dynamic programming calculates the best score for every single square, one by one. Global alignment forces the computer to align the entire length of both sequences, from the first letter to the last. It builds a scoring matrix by evaluating rewards for matches and penalties for mismatches and gaps at every position. Finally, it traces back from the end to the beginning to reconstruct the mathematically perfect path. This is crucial when comparing sequences of similar lengths. Let us look at the mathematical engine behind this algorithm. This formula calculates the value of each cell in our scoring matrix. F of I, j is the maximum alignment score at the current position. The function S represents the substitution score, which gives us a positive number if the characters match and a negative number if they mismatch. The variable D is the gap penalty, a negative number subtracted when we introduce a gap. The formula looks at three previously calculated cells: the diagonal, the top, and the left. It calculates the score for arriving from each direction and uses the max function to choose the highest possible value. This guarantees we always take the best possible path. Now that we have our formula, let us use it to align the short DNA sequence AT with AG. We are given a match score of +1, a mismatch of -1, and a gap penalty of -1. First, we initialize the matrix edges with accumulating gap penalties: 0, -1, -2. Now we calculate the first cell for A versus A. We check all three directions. Coming from the diagonal, we add our match score of 1 to 0, giving us 1. Coming from the top or left means adding a gap penalty, which gives us -2. The maximum of these three options is 1, so we write 1 in that cell. Continuing our example, we move to the bottom right cell to compare T versus G. We look at the diagonal score coming from our previous cell, which is 1. Since T and G do not match, we add our mismatch penalty of -1. 1 minus 1 equals 0. Checking the top and left paths would give us even lower scores because of gap penalties. Therefore, the final score at the bottom right is 0. To find the alignment, we trace back the path that gave us these highest scores, which goes straight up the diagonal. This tells us the optimal global alignment is A aligned with A and T aligned with G, requiring no gaps. Global alignment is perfect for similar sequences. But what if we are looking for a tiny needle in a massive haystack? Suppose you want to find one specific paragraph in a 1,000-page encyclopedia. You do not care about the rest of the book. This is where local alignment, using the Smith-Waterman algorithm, comes in. Instead of forcing sequences to line up from beginning to end, it scans them to find the most similar subsections, completely ignoring the unrelated junk data around the match. This is incredibly useful when sequences have vastly different lengths. It is the ultimate tool for finding a specific functional domain, like a DNA binding motif, hidden within a massive, otherwise unrelated protein. The formula for Smith-Waterman is almost identical to Needleman-Wunsch, but with one game-changing addition. Look closely at the max function. The variables mean the exact same thing. F is the current cell score, S is the substitution score, and D is the gap penalty. However, notice the 0 as the very first option. This means if all the other calculations result in a negative number, the cell simply takes the value of 0. This 0 acts as a fresh start. It prevents bad alignments from dragging the score down into negative infinity. When we trace back, we do not start at the bottom right. We start at the highest number in the matrix and stop when we hit a 0. Let us see how this zero reset works in practice. Imagine aligning the short sequence, G, C, C, against a much longer sequence, A, T, C, G, C, C, G, T, A. If we used global alignment, the extra letters at the beginning and end would create massive gap penalties, ruining the score. But with Smith-Waterman, as the matrix calculates the unrelated ATC part, the scores repeatedly drop below 0 and are instantly reset to 0. The matrix stays at 0 until it hits the matching GCC section. The score climbs to 1, then 2, then peaks at 3. The trailing letters drive it back to 0. We trace back from the 3 until we hit a 0, perfectly extracting our hidden motif. Dynamic programming guarantees the mathematically perfect alignment, but it has a major flaw. It is too slow. Today, biological databases contain billions of sequences. Running Smith-Waterman against an entire database would take months on a supercomputer. This is why we use heuristic methods like BLAST and FASTA. Think of a heuristic as a computational shortcut. It sacrifices a tiny bit of mathematical accuracy for a massive, exponential increase in speed. Instead of calculating a full matrix, these tools quickly scan for short, exact matches called words or k-mers. Once they find a perfect word match, they use it as an anchor and attempt to extend the alignment outward. This allows BLAST to search billions of sequences in mere seconds on a standard laptop. Because BLAST uses shortcuts, it cannot just give you a score. It must tell you how statistically significant that score is. It does this using the e-value formula. E is the expect value, which represents the number of times we would expect to see an alignment score of S purely by random chance in a database of this size. The variable m is the length of your query, and n is the massive length of the entire database. K and lambda are simply statistical constants. The most important relationship here is between the alignment score S and the e-value. As your alignment score increases, the exponential term becomes very small, driving the e-value closer to 0. A lower e-value means a highly reliable match. Let us interpret a real-world BLAST search using this formula. Suppose you sequence a new piece of DNA in the lab and run it against the entire human genome database. The search finishes in seconds and returns a match with an alignment score of 200 and an e-value of 10 to the power of negative 50. What does this mean? An e-value of 10 to the negative 50 is practically 0. It tells you that the probability of finding a match this good by pure random chance is astronomically low. Because it is so close to 0, you can confidently conclude that your new DNA sequence is genuinely related to the one in the database, likely sharing a common evolutionary ancestor. We have covered a lot of ground today, so let us summarize the most important ideas. First, sequence alignment is the foundational tool for identifying evolutionary and functional relationships in biology. We saw how the dot matrix method gives us a quick, visual way to spot large-scale mutations. Then we explored dynamic programming. The Needleman-Wunsch algorithm guarantees the optimal global alignment for sequences of similar length, while the Smith-Waterman algorithm uses a zero reset to find local, hidden motifs in vastly different sequences. Finally, we learned that for massive modern databases, heuristic tools like BLAST use word-matching shortcuts and statistical e-values to deliver highly accurate results in seconds. You now understand the computational engines that drive modern genomic research.